One of the most fundamental attributes of any living creature is the ability to bring forth live young ones of its own kind for prosperity and continuity of generations over time. Welcome to the next chapter. It is reproduction. Now, reproduction is the process by which mature individuals produce offsprings of their own kind. It is an essential characteristic of all living organisms. There are two types of reproduction which are based on the ability of the cell to divide and form new daughter cells: sexual and asexual reproduction. Sexual reproduction involves the fusion of the male and female gametes to form a zygote. The fusion of the nuclei of the male and female gametes is known as fertilization. Asexual reproduction does not involve gametes whatsoever, but instead parts of the mature organisms may develop to new individuals. Reproduction is an important characteristic of all living organisms. Why? Because it ensures procreation. Reproduction sustains or perpetuates the species so that it does not become extinct. Since no living organism lives forever, every species must produce new individuals in order for the species to continue. It also improves the quality of the species. It allows for mixing of genetic materials, which leads to variations amongst individuals in a species. This is observed in sexual reproduction, and the variations form the basis of adaptability to their habitats and to the changing environmental conditions. Now, life in living organisms starts as a single cell, a spore or a zygote, and through cell division the number of cells increase. In multicellular organisms, the cells divide into two and continue that way while still attached to the neighboring cells. In unicellular organisms, the cells divide into separate daughter cells. But what is important to note is that after division, each cell attains autonomy and possesses the machinery and mechanisms to divide again. So we have a kind of exponential increase in the number of cells. But the process of cell division is not as simple as it appears. It involves signals that initiate the process when needed and signals to terminate the process. It is the breakdown of this regulatory mechanism that often leads to development of tumors. Before a cell divides, all its contents must be duplicated to ensure that each daughter cell gets all the materials and organelles needed to carry out physiological processes on its own. A very important component is the DNA. So cell division starts in the nucleus, that is the chromosomes, and then the cytoplasm. So let us first look at the chromosomes. And the DNA of a cell is packaged into long thread-like structures which we call the chromosomes. Normally each cell has a fixed number of chromosomes, and each chromosome is made up of two parallel strands called chromatids. Each pair of chromatid is connected at a point by a structure called the centromere. Chromosomes are present in the nucleus all the time, but they only appear or become visible when viewed under a microscope during cell division. In body cells, chromosomes occur in pairs. Each pair of chromosomes has a characteristic length. The member of each pair is called a homologous chromosome, and this means that the chromosomes are alike in appearance, although their genetic composition may be different. Now, during sexual reproduction, each parent contributes one of the chromosomes from the homologous pair. Along the length of the chromosome is a series of structures called genes, and they are too small to be seen even with a powerful microscope. Genes determine the characteristics of the cells and its progeny. Genes are made up of a protein chemical that is called the DNA, which contains coded information or instructions that dictate the characteristics of the offspring. For example, whether male or female. Young cells of an organism divide actively and grow before they differentiate into tissues and organs. The ability of cells to divide is located in specific areas where the cells are unspecialized, and these include the meristematic cambium cells, meristematic apical cells in woody plants, and in the malpighian layer in the mammalian skin. The sequence of events leading to cell division in plants and animal cells are basically the same, and there are two types of cell divisions: mitosis and meiosis. Now, before we start to look at the process of mitosis, it is important we mention something to do with the cell cycle. In most eukaryotic cells, division occurs every 24 hours, and mitosis is only a small portion of the cycle, taking only around 80 minutes. During all the other time, the cell undergoes preparation for replication, and this is called the interphase stage. First, the cell grows to nearly its full size and performs other biochemical functions. Then the DNA in the nucleus is replicated to produce sister chromatids. After this, the cell completes its growth with the DNA replicated and two centrioles appearing in the cytoplasm. And now the cell is ready for mitosis. During mitosis, a cell will divide to form two nuclei and then divide into two cells during what we call cytokinesis. Mitosis divides into two the duplicated DNA of a cell. It is split into distinct stages. The first one is prophase, where the DNA condenses, organizes, and chromosomes appear. Then we have the prometaphase, where microtubules attach to the chromosomes. Then follows the metaphase, where chromosomes align. The chromosomes then separate in anaphase. Finally, nuclear envelopes appear around the two sets of chromosomes. After mitosis, two cells are formed by the process of cytokinesis. During the first stage, the prophase, the classic chromosome structures become visible, and this occurs through a condensation process. At the same time, protein strands called microtubules or spindle fibers appear from the centromeres in animal cells. The nucleolus disappears. A short phase called prometaphase begins, where the nuclear membrane is broken down. At the same time, microtubules or spindle fibers are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Then comes metaphase. Here, sister chromatids are lined along the center of the cell. Now the sister chromatids are ready to be separated. The separation occurs in anaphase by shortening of the microtubules. Additionally, the poles of the cell moves further apart, causing increased separation of the sister chromatids. At the end of anaphase, the chromosomes have moved to the end of the cells. Telophase is the final stage of mitosis. It's here that the components of the new cells begin to appear. The spindle fibers are broken up. A new nuclear membrane surrounds each set of chromosomes, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The splitting of the cell is all that remains. This occurs by cytokinesis. A contractile ring compresses the cell, dividing it into two nearly equal halves. By this time, the organelles in each cell have been replicated. This includes the mitochondria, Golgi bodies, and RAF-ER. Plant cells also have chloroplasts. Now each cell is autonomous and ready to start the preparation for division, and the cycle continues. You notice that sister chromatids are duplicates of each other, so they are similar and identical in every sense. They carry the same genetic material. So when they are separated in mitosis, the daughter cells have exactly the same genetic material. So in mitosis, there is preservation of genetic material. Thus, all body cells except the sperm and eggs have the same genetic material because they all started as one cell. So exactly what is the significance of mitosis? Now, mitosis is the type of cell division that takes place during the growth of an organism. For example, in development of a fertilized egg into an adult human being. The fact that these divisions are mitotic means that all the cells of the body have a diploid or a double number of chromosomes, also referred to as 2N constitution. 23 pairs in the case of human, 4 pairs in Drosophila, 7 pairs in garden pea, and 20 pairs in a mouse. Mitosis is also the basis of asexual reproduction. It is the process that occurs when amoeba undergoes binary or multiple fission, or when a bud develops in yeast. Mitosis ensures that the chromosomal constitution and hence the genetic constitution of the offspring is the same as the parents. Meiosis. Meiosis occurs in special cells called germ cells within the gonads of males and females. For successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These are termed as meiosis 1 and meiosis 2. Although meiosis and mitosis look similar, there are few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, whereas meiosis results into 4 haploid daughter cells. But just like in mitosis, meiosis begins after the cell has successfully completed the interphase part of the cell cycle. During interphase, the DNA is replicated, producing 2 copies of each chromosome, called sister chromatids. The paired central in the cytoplasm duplicate and begin extending microtubules that form the spindle fiber. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1. Up to this point, the cell looks similar to that in mitosis, but two events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous chromosomes lie side by side in a process called synapsis, forming a bivalent. Remember, homologous chromosomes share similar but not necessarily identical genes. What that means is that, for instance, the chromosome may carry the genes for color vision, but one chromosome carries the gene for colorblind and the other for normal vision. Sister chromatids are identical themselves. Once this structure has been formed, the second event called crossing over can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 ends with the disintegration of the nuclear envelope as the duplicated centromere pairs move to the opposite poles of the cell. They extend their spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired central in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kineticals. Here, another key difference between meiosis and mitosis occurs. Because of synapsis and crossing over, homologous chromosomes remain aligned so that each pair of sister chromatids is attached to only one pole of the microtubule. In mitosis, each sister chromatid is attached to a spindle fiber originating from the opposite poles. In metaphase 1, the bivalents arrange in the middle, what we call the metaphase plate. Due to independent assortment, this alignment is random, as shown here, and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move towards the opposite poles. Meiosis 1 ends with telophase 1 when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the end product of meiosis 1 is two haploid daughter cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles duplicate again and begins to move to the opposite poles of each cell. In prophase 2, the sister chromatids condense, the spindle fibers begin to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetical microtubules, with sister chromatids attached to the opposite poles. The sister chromatids align along the metaphase plate in metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move towards the pole. The entire process ends with telophase 2 as all the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and a cleavage furrow separates the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either a sperm or an egg. These fuse in fertilization to form a zygote which will grow into a child. The child receives its half chromosomes from the mother and half from the father. Because men and women produce millions of gametes and the selection of gametes in fertilization is random, This contributes to genetic diversity, and this explains why children are not exactly like their parents. But why are siblings not identical? The diversity comes from several sources. In Prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing genetic diversity of individual chromatids. Again, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the index power of n. So if an organism has 3 chromosomes, that is, n is equal to 3, it can produce 8 unique gametes. For humans, where n is equal to 23, there are 2 to the index power of 23 unique gametes formed. This is equivalent to over 1 million different possible combinations. The combination of crossing over, independent assortment, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents nor siblings. So exactly what is the significance of meiosis? Meiosis occurs during the formation of gametes or the sex cells such as eggs and sperms. These contain half the number of chromosomes as the parent cell in those organisms which carry out sexual reproduction. Meiosis helps to restore a constant diploid chromosomal constitution in a species after fertilization. During fertilization, the nuclei of the 2 gametes fuse to produce a zygote, which has a fixed number of chromosomes for each species. This represents the diploid condition. If meiosis never occurred, fusion of gametes would result in doubling of the chromosomes for each successive sexually reproduced generation. Meiosis also provides opportunities for new combinations of genes to occur in the gamete cells. This leads to genetic variation in the offspring produced by fusion of gametes. The other type of reproduction is asexual. Asexual reproduction is the production of offspring from single organisms without fusion of gametes. Meiosis is not involved and the offsprings are identical to the parents. There are 3 types of asexual reproduction: binary fission in amoeba, spore formation in Rhizopus species, budding in yeast. Binary fission. Binary fission is the simplest form of asexual reproduction. This method involves splitting of the original cell into 2 similar daughter cells. The fission occurs when the organism attains the maximum size. Binary fission is a complex process that includes internal reorganization, division of the nucleus, and cytoplasmic division. Briefly, the nucleus divides into 2, then the cytoplasm divides to form 2 daughter cells. Binary fission is common in amoeba, plasmodium, and bacteria. Spore formation or sporulation. A spore is a small reproductive unit which is usually microscopic and unicellular, containing a small amount of cytoplasm and a nucleus. Spores are produced by bacteria, most fungi, and some groups of green plants such as mosses and ferns. Spore formation in rhizopas. Rhizopas is a saprophytic fungus which grows on substrates like bread, rotting fruits, and other decaying organic matter. The vegetative body is called the mycelium, which consists of many branched threads called the hyphae. The horizontal hyphae are called stolons. The hyphae which grow vertically are called sporangiophore. The tips of the upright hyphae swell up to form the sporangia, the spore-bearing structures. Inside the sporangium, the nuclei divide meiotically, producing daughter cells that contain a haploid number of chromosomes. The daughter nuclei developed into spores by the incorporation of cytoplasm around each nucleus and a wall around the cytoplasm. The sporangium contains many spores. As it matures and ripens, it turns black in color. When fully mature, the sporangium wall bursts and releases the spores which are dispersed to restart a fresh generation of growth and development. Spore dispersal may be by wind, by an insect, or mechanically being scattered by animals. Budding in yeast. Budding is a form of asexual reproduction in which a new individual is produced as an outgrowth or bud of the parent and is later released as a self-supporting identical copy of the parent. It takes place in unicellular fungi such as yeast. The yeast cell, under conditions that are favorable, that is plenty of sugar, moisture, oxygen and optimum temperature, reproduces asexually by budding. In budding, a small area of the cell wall of a parent cell softens and forms a projection of a bud which bulges outwards. This is followed by the division of the nucleus into two. One of the nuclei moves into the new bud. The bud then grows in size and forms new cell organelles. Sexual reproduction in plants. Sexual reproduction is as significant in plants as it is in animals, where asexual methods are virtually unknown. In flowering plants, the flower is the reproductive organ, which is a specialized shoot consisting of a modified stem and modified leaves. Typically, the stem portion consists of a pedicel, the flower stalk, and a receptacle, to which are attached the modified leaves. After pollination followed by fertilization, the ovary of the flower forms the fruit which encloses the seeds which can later germinate to form a new plant. Now we first look at the structures and function of a flower. A flower is made up of a flower stalk called the pedicel, the apex of the flower stalk called the receptacle, to which are attached the four groups of floral structures, namely the calyx on the outside, corolla, the colored parts, the male parts called the androecium, and the female parts called the gynoecium or the pistil. The calyx is made up of sepals, which are usually green. The calyx protects the inner parts of the flower, especially during the bud development. In some flowers, there may be another ring of sepal-like structures just beneath the calyx called the epicalyx. The corolla is made up of petals which are brightly colored, large and conspicuous, especially in insect-pollinated flowers where they attract insects for pollination. Androecium is the male part of the flower and is made up of one or more stamens. The stamen consists of two parts. The long stalk-like structure, the filament, which bears the anther at its tip. The other consists of 4 pollen sacs, which contains pollen grains. These contain the male gametes. The stamen may be either fused, forming a staminal tube, as in hibiscus, or it may be free, as in a crotalaria flower. Gynoecium, or the pistil, consists of carpels that form the female reproductive organ of the flower. It may consist of one, a few, or many carpels. A carpel consists of the ovary, the style, and the stigma. The ovary contains ovules. Next is pollination, and pollination is the transfer of pollen grains from the anther to the stigma. When the transfer is from the anther of one flower to the stigma of another flower but of the same species, it is called cross-pollination. If the transfer of the pollen grains is from the anther to the stigma of the same flower, it is referred to as self-pollination. The structural arrangements and the differences in time of ripening of the male and the female parts of the flower may necessitate the intervention of some agents to aid in the transfer of pollen grains to the stigma for fertilization. Agents of pollination include wind and insects. For pollination to occur, flowers are structured in such a manner as to enable the appropriate agent to carry out pollination without hindrance. Insect pollinators include bees, butterflies, and mosquitoes, which usually visit such flowers for the purpose of getting nectar. Wind pollination occurs mostly in plants of the grass family like maize, wheat, and oats. Insect-pollinated flowers are also called entomophilous flowers like the hibiscus. The flowers are large, conspicuous, with brightly colored petals, bracts, or inflorescence. This attracts the insects. The flowers are also scented to produce nectar. These are for attraction of the insects which come to obtain the nectar for their food. Fertilization in flowering plants. Fertilization is the fusion of the male and female gametes to form a zygote. In flowering plants, a special kind of fertilization called double fertilization is exhibited. After pollination, the pollen rests on the stigma. The pollen has 2 layers. The outer layer is called the exine. The inner layer is thin and is known as intine. At some places, the exine is thin, and these areas are known as germ pores, through which the pollen tube grows. The pollen grain has 2 cells. The smaller cell is called the generative cell, which will divide to form 2 male gametes. The larger cell is known as the tube cell, which will give rise to the pollen tube. The embryo sac has 2 cells towards the chalaza, known as the antipodals. There are 2 nuclei in the middle called the polar nuclei. 3 cells are found towards the micropyle. The middle larger cell is called the egg, and the 2 smaller cells are called the synergids. The egg and the 2 synergids will form the egg apparatus. A pollen tube emerges from the germ pore. The haploid generative cell divides mitotically to form 2 male gametes. The pollen tube grows down the style to the embryo sac, taking along with it the male gametes. The pollen grains usually adhere to the stigma as a result of the stigma cells secreting a sticky substance which also stimulates the pollen grain to germinate, sending down its pollen tube. The growth of the pollen tube into the stigma through the style to the ovary is by pushing its way between the cells where it gets its nourishment from the surrounding tissues. The process is quite rapid and takes place in a matter of minutes. The tube enters the embryo sac through the micropyle. The first male gamete fuses with the egg to form a diploid zygote which develops into an embryo. The second gamete fuses with the two polar nuclei to form a triploid endosperm nucleus, which will develop into an endosperm. The fusion of the three nuclei is known as triple fusion. Endosperm will divide later on to form nutritive tissue, which will provide nourishment to the developing embryo. The double fusion of the male gametes with the eggs and the polar nuclei is called double fertilization. Seed and fruit development. Now, seed and fruit start developing after fertilization has taken place. During the double fertilization we saw, one of the sperm cells fertilizes the egg cell, forming a diploid zygote which will grow into a seed embryo. Then in the second fertilization, the other sperm cell fuses with the two polar nuclei, forming a triploid cell. Its nucleus then divides to form a large multinucleated cell. Eventually, membranes and cell walls take shape between the nuclei, creating the endosperm, which in the mature seed is the solid nutrient-rich material that nourishes the plant's developing embryo. In many plant species, fruits start to develop while the seeds are being produced. Most fruits develop when the wall of the ovary known as pericarp thickens as a result of storing sugary and other nutrient-rich materials. A fruit is an excellent way for plants to disperse seeds because fruits are favorite foods for animals which carry the seeds in the alimentary canal and dump them further away as fecal waste where these seeds germinate. Now the seeds develop inside the fruit, forming different types of placentations as shown on your screen. Sexual reproduction in animals. Sexual reproduction involves the fusion of gametes, and in animals, two individuals are involved, male and female. The males produce gametes known as sperms, and the females produce gametes known as ova. These gametes are produced by special organs known as the gonads, the testes which produce sperms, and ovaries which produce the ova. The sperm fuses with the ovum to form a zygote, a process called fertilization. The gametes are haploid, and the zygote so formed is a diploid. Fertilization may occur internally or externally. External fertilization occurs in animals such as amphibians and fish. This is only possible in water. During this time, the female lays eggs in water and the male sheds sperms over these eggs to fertilize them, resulting in fertilization outside the body. In this kind of fertilization, many eggs are released to increase the chances of survival, since the fertilized eggs could be attacked by bacteria or eaten by some other animals. During the mating season, the males croak mainly at night to attract females. While mating, the male mounts the female and sheds sperms as the female lays eggs. These activities increase the chances of fertilization. The eggs are laid in long strands of slippery jelly-like substance which provides protection to the eggs. At the same time, the jelly prevents predators from feeding on the eggs. This substance separates the eggs from each other, allowing for good aeration. It also attaches the eggs to the water plants and also makes the eggs to be buoyant. Internal fertilization, on the other hand, occurs in reptiles, birds, and mammals. Fertilization occurs within the body of the female. Generally, the number of eggs produced by the females are fewer than those produced by animals where external fertilization occurs, and this is because there are higher chances of fertilization since the sperms are deposited into the female's body. Furthermore, protection of the gametes and fertilized eggs is higher. Fertilization in mammals occur internally, and after fertilization, the fertilized egg is laid or develops within the female's body in a special structure called the uterus. Reproduction in human beings. Structure and functions of the male reproductive system. The male reproductive system consists of a pair of testes enclosed in a loose sheath called the scrotum. From the testes leaves the vas deferens or sperm duct. We also have three glands, namely the seminal vesicle, prostate gland, and the corpus gland. The vas deferens leads to the urethra, which is a continuous way into the penis. The testes lie outside the abdominal cavity in the scrotal sac. The position of the testes outside the body is significant as it provides a cooler environment for sperm production. Sperms develop best at a lower temperature than that of the body. The testes originate in the abdomen from the embryonic tissue, but in mammals, they later move down into the scrotal sac. The testis is made up of several highly coiled tubules known as the seminiferous tubules. The lining of these tubules consists of actively dividing cells which give rise ultimately to sperms. These cells are the Leydig cells, also known as the interstitial cells, which secrete the male hormone testosterone. Sertoli cells support sperm development. The smooth muscle creates peristaltic movements which propels the sperms through the seminiferous tubules. Now, once the sperms have been made, they leave the seminiferous tubules. So here are the sperms in the seminiferous tubules, which have been made by the Sertoli cells and released into the lumen of the seminiferous tubules. The sperms then travel through this little region here called the rete testis and into the efferent ductules. This is the point that the sperms exit the testis. From here, they enter the epididymis for storage. During ejaculation, the sperms leave the epididymis and into the vas deferens. The vas deferens connects with the seminal vesicle to form the ejaculatory duct. At this point, the sperms are mixed with the fluid from the seminal vesicle. The ejaculatory duct penetrates through the prostate gland to join the urethra. The sperms end up in the urethra, which receives substances from the bulbourethral glands or the corpus gland. The three accessory glands include the seminal vesicles, prostate gland, and the corpus gland. The seminal vesicle is a blindly ending sac which also opens into the urethra at this region. It provides an alkaline fluid which contains nutrients for the spermatozoa. Near this point is situated the prostate gland, whose secretion is alkaline to neutralize the vaginal fluids. It also activates the sperms. Just below the prostate gland on either side of the urethra lies the corpus gland, which secretes an alkaline fluid that neutralizes the acidity along the urethra. All the fluids combine with the spermatozoa to form the semen. Structure and function of the female reproductive system. Now on your screen is the structure of the female reproductive system. There are two ovaries which are cream-colored oval bodies measuring between 3 and 4 centimeters long. They are found in the lower part of the abdomen below the kidneys. Ovaries are held in position by membranes and blood is supplied to them by the ovarian artery and removed by the ovarian vein. The ovaries produce eggs and female hormones. An oviduct is a tube with an expanded funnel-shaped opening which conducts ova released by the ovary to the uterus. It is lined with cilia which waft the ovum towards the uterus. The movement of the ovum is also aided by the contractions of the smooth muscles of the tubal wall. Fertilization occurs in the oviduct. The uterus is found in the lower abdomen. It is in this structure where the embryo develops. The uterus is capable of expanding as embryo grows and can return to original size of about 8 cm in length after birth. This is possible because its walls are muscular. Its muscles are also important at the time of birth as they aid in the expulsion of the fully developed fetus. The uterine wall is lined by an endometrium for implantation and nourishing of the embryo at its initial stages. The uterus is kept in position by ligaments and is supported by the bladder and the muscular floor of the pelvis. At the lower narrow end of the uterus, which leads into the vagina, is the cervix. The cervix has a ring of muscles that separate the uterus from the vagina. It secretes a plug of mucus which prevents the entry of pathogens into the uterus during pregnancy. The vagina is the copulatory canal. It is also called the birth canal. The vagina, together with the urethra, opens to the outside at the vulva. Within the walls of the vulva are the vestibular glands which release mucus when the female is sexually aroused, which helps to lubricate the vagina during intercourse. Now the process of fertilization. But for proper discussion of fertilization, it is necessary to understand the structure of the male and female gametes. The sperm consists of an ovoid head, a short neck, a middle piece, and a tail. The head contains a large nucleus. The nucleus carries the genetic material which is haploid in number. At the tip of the head is an acrosome which contains lytic enzymes. These enzymes digest the membrane of the egg for the sperm to penetrate and effect fertilization. The short neck contains centrioles. The middle piece is packed with a large number of mitochondria, and these mitochondria produce energy necessary for propulsion of the sperm to reach the egg. The tail propels the sperm forward by its side-to-side lashing action. Unlike the production of sperms in males, which only begins at puberty, the process of egg formation begins in the ovary of the fetus before birth. At birth, there are about 70,000 potential egg cells in the ovaries of a baby girl. Of these, about 500 of them may develop into ova during puberty. This involves the process of meiosis. A mature human ovum is spherical in shape with a diameter of about 0.2 millimeters. It consists of a large haploid nucleus surrounded by a nuclear membrane. The nucleus is within the cytoplasm enclosed by a plasma membrane. Another membrane, the vitelline membrane, surrounds the plasma membrane. Now, fertilization is the process whereby the nucleus of a male gamete fuses with the nucleus of a female gamete to form a zygote. This occurs in the upper part of the oviduct after copulation. The sperms are drawn up by suction through the cervix into the uterus, from where they swim up to the oviduct using their tails. Although a single ejaculation contains millions of sperms, only one will fertilize the ovum. As the ovum moves down the oviduct, it releases chemical substances which have to be neutralized by those produced by the acrosome before the sperm penetrates the ovum. When the sperm comes into contact with the egg, the acrosome bursts open and releases the lytic enzymes which dissolve the egg membrane. The acrosome then turns inside out, forming a fine filament that is used to penetrate the egg. The head of the sperm enters the ovum and the tail is left outside the ovum. After fertilization follows implantation, and this is the attachment of the blastocyst to the wall of the uterus by the villi. Normally, implantation takes place in the uterus. After fertilization, the zygote undergoes several mitotic divisions as it moves down the oviduct. Its movement is aided by the cilium movement and the contractions of the smooth muscles along the oviduct. The movement of the zygote from the oviduct to the time it gets implanted lasts for about 7 days. By the time it reaches the uterus, it has formed a hollow structure of several cells called a blastocyst. In the uterus, the blastocyst develops finger-like projections called villi, which grows into the wall of the uterus, that is the endometrium. The villi, together with endometrium, develops into a special organ called the placenta. Upon implantation, the blastocyst becomes an embryo. Occasionally, the zygote fails to move down the oviduct to the uterus, but instead gets implanted into the walls of the oviduct, where it develops, and this condition is referred to as ectopic pregnancy, which is fatal if carried to term in most cases. During implantation, the blastocyst differentiates into 3 layers, namely the chorion, amnion, and allantois. The chorion, which is the outermost layer, develops finger-like projections called the chorionic villi. These villi grow into the endometrium. During the early stages of embryo development, the villi form the sites for material exchange between the embryo and the maternal blood vessels in the uterine wall. The amnion surrounds the embryo, forming an amniotic cavity within which the embryo lies. The amniotic cavity is then filled by a fluid secreted by the amnion called the amniotic fluid. This fluid provides a fluid environment which later suspends the fetus, providing it with support. It also acts as a shock absorber and hence protecting the fetus against mechanical injury. The chorionic villi, allantois, together with the endometrium form the placenta. The embryo is attached to the placenta by a tube called the umbilical cord, which increases in length as the embryo develops. When the placenta is fully formed, the embryo becomes a fetus and this lasts about 3 months of pregnancy. The placenta is a temporary organ found only in placental mammals and is the only organ in animals composed of cells derived from two different organisms, that is the fetus and the mother. It is a point of close association between the maternal and fetal circulations and facilitates the transfer of nutrients and metabolic waste products between the fetus and the mother. The placenta selectively allows some substances to pass through and not others. Drugs, alcohol, and some chemicals from cigarette smoke pass through the placenta, and hence pregnant mothers are advised not to take alcohol nor smoke excessively. If she has to take any drug like aspirin, it has to be under the instructions of a doctor. Now, the fetal blood does not mix with the maternal blood in the placenta. The exchange of materials occur across the sinus in the uterine wall and the capillary systems of the fetus across the intercellular space by diffusion. This is because there is no direct connection between the fetal blood system and that of the mother. If the two systems were directly connected, the delicate blood vessels of the fetus would burst due to the high blood pressure in the maternal circulatory system. During pregnancy, the placenta takes over the role of producing hormones, estrogen and progesterone. The period between conception and birth is termed as gestation. The gestation period varies in different mammals. In mice, the period is about 22 days, 30 days in rabbits, and 9 months in humans. In large mammals like the elephant, it is about 18 months. 2 weeks old, the 3 embryonic membranes, that is allantois, amnion, and chorion, have already formed. The embryo now appears as a bump in the uterine wall. The embryo then differentiates into tissues and organs. By the end of the 3rd month, the heart and blood vessels forming the fetal circulatory system are fully developed. The head region, which includes the nose and eyes, is also well developed. The other regions which are formed by this time are the spinal cord and the tail. The limbs also show early signs of development. Within the next 3 months, the limbs, alveoli, and the nostril are well developed and the fetal movements can be felt. By the end of the 9-month period, the fetus head is directly above the cervix and all the organs and systems are fully developed such that they are functional at the time of birth. Normally, birth occurs after the 9-month period. If birth occurs before the completion of 6 months, the baby will not survive, and this is referred to as miscarriage. In the case where fetal development is interfered with, either chemically or physically, such that the fetus is expelled, the condition is called abortion. However, if birth occurs after the 7th month but before full term, such is referred to as premature birth. A premature baby can survive if it has attained a certain weight, or else it will require specialized medical care. In hospitals, premature babies are cared for in incubators. The onset of birth is signaled by the reduction in progesterone level in the mother's blood, which allows the maternal posterior pituitary gland to release the hormone oxytocin. Oxytocin removes the inhibitory effect on contraction of the myometrium, whose muscles start contracting. The release of oxytocin occurs in waves during labor and provides the force to expel the fetus from the uterus. The onset of contraction of the myometrium, called labor pains, are accompanied by the dilation of the cervix, the rupture of the amnion and chorion, releasing the amniotic fluid through the cervix and the stimulation of stretch receptors in the walls of the uterus and cervix. The contraction of the uterus wall follows. Uterine contraction spreads down over the uterus and thus pushing the fetus downwards, heads first through the widened cervix and birth canal, and the baby is born. After birth, the umbilical cord is tied and cut in order to separate the baby from the placenta. The placenta is expelled later as afterbirth. Next, we'll look at the role of hormones in human reproduction, and specifically in the menstrual cycle, better known as the ovarian cycle. Now, in females, gamete production is a cyclical activity with a periodicity of approximately 28 days. It is called the menstrual cycle and is under the control of hormones from various sources. The main players in the female reproductive cycle are the pituitary gland, the ovary, and the uterus. The activities are closely coordinated. Each month, one or the other ovary releases a single egg, an event known as ovulation. It is brought about by a series of complex interactions between the pituitary gland, the ovaries, and the uterus. The pituitary gland is itself under the control of a small area of the brain known as the hypothalamus. A new menstrual cycle begins when the nerve cells of the hypothalamus secrete a hormone called gonadotropin-releasing hormone, GnRH, into the network of the blood vessels which surround the pituitary gland. Stimulated by the pulses of GnRH, cells of the pituitary secrete another hormone, follicle-stimulating hormone, FSH. FSH travels in the bloodstream, reaching the ovaries. There, it stimulates the formation and growth of an ovarian follicle in one of the ovaries. The follicle consists of an egg, a number of surrounding cells which secrete estrogen hormone and fluid. FSH helps the egg to mature and prepares it for release. As the follicle matures, the hypothalamus increases secretion of GnRH. This in turn stimulates the pituitary to release yet another hormone called the luteinizing hormone, LH, which acts on the ovary. Towards the middle of the cycle, there is a peak in the blood levels of LH. This acts as the trigger for ovulation. Within minutes of its release, the egg is guided by suction through the opening of the fallopian tube, starting on a journey which will take approximately 5 or 6 days as it passes through the tube and finally reaches the cavity of the uterus. Meanwhile, after the follicle ruptures, it is converted into this yellowish body called the corpus luteum. Cells of the corpus luteum secretes the hormone progesterone, which brings about important changes in the linings of the uterus, preparing it for possible pregnancy. In fact, the lining of the uterus, known as endometrium, undergoes changes in response to the hormonal levels during the cycle. In the first phase, known as follicular phase, the developing follicle secretes increasing amounts of estrogen hormone, which encourages regeneration of the endometrium. After ovulation, there are important changes in the endometrium aimed at making it suitable to release a fertilized egg. These changes are brought about by secretion of progesterone from the corpus luteum. The secretion of progesterone is maintained for several days, but if fertilization does not occur in those days, the corpus luteum degenerates and falling levels of progesterone and estrogen trigger the shedding of the endometrium and the menstrual flow, and the cycle starts all over again. But if the egg is fertilized, no menstruation occurs as the corpus luteum continues to function, secreting progesterone within the first 3 months of pregnancy. Thereafter, numerous changes occur to support the developing embryo and maintain the pregnancy. The changing levels of the various hormones involved in the menstrual cycle can be summarized as shown on this graph.
Without many of these discoveries and inventions, few of us would be alive today. Welcome to WatchMojo.com, and today we're counting down our picks for the top 10 greatest medical breakthroughs in history. Number 9. The birth control pill. Both the medical and social spheres of human life were significantly affected with the introduction of the birth control pill in the 1960s. By preventing unwanted pregnancies, the contraceptive displayed the great potential of medicine and gave women power over their own lives. The pill also helped ensure eventual salary parity between the sexes and indelibly aided the feminist revolution.